Bismillahirrahmanirrahim. In this video, the process of remodeling is discussed. These are the references for this video. The tissue remodeling phase of the repair is the longest phase of repair, and it follows the inflammatory and proliferative phases. The key cellular events in this tissue remodeling phase are decline of inflammatory cells, capillary formation is completed, and fibroblasts first increase and then decline. The main enzymes that are responsible for extracellular remodeling are matrix metalloproteinases, cathepsins that come from neutrophils, and serine proteases. The enzyme localization, the matrix metalloproteases, and ADAMs, that is the disintegrin and metalloproteinases, are highly localized to the tissue region, while neutrophilic enzymes are evident very early in the phase and they are distributed diffusely. The key protease superfamily that is involved in this process is MET-zinc-sins. These are zinc-dependent catalytic site enzyme. These include matrix metalloproteinases, ADAMs, a disintegrin and metalloproteinase, and ADAMTS, that is a disintegrin and metalloproteinase with thrombospondin motif. The function of medicines is to regulate the extracellular matrix turnover during development, wound repair, and tissue remodeling. The protease regulation is controlled by inhibitors of metalloproteinases that are termed as TIMPs or tissue inhibitors of metalloproteinases. There are about 25 matrix metalloproteinases are identified, and they are responsible for degradation of the extracellular matrix to enable the migration of the cell, remodeling, and wound healing. Their other functions are to modulate the signaling of the cell, regulate apoptosis and proliferation, or activate or inactivate the bioactive molecules. The forms of MMPs are secreted one, and they are termed as MMP1, 2, etc. And other are membrane type MMPs that are abbreviated as MTMMPs, MT membrane type matrix metalloproteinases. The anchoring mechanism of these membrane type is by the small cytoplasmic tail, and there is glycophosphoinositol anchor especially for MT4 MP and MT6 MMP. They are synthesized as zymogen and present as inactive proenzymes and activated by MMP3, 14 and serine proteases. Then it is discovered that many substrates are cleaved by multiple MMPs that suggest their regulatory importance. So the substrates for these matrix metalloproteinases are clotting factor, extracellular matrix protein, latent growth factor and growth factor binding protein, receptors for matrix molecules and cell-to-cell adhesion molecules, immune system components, other MMP, other proteinases and proteinase inhibitor, and various chemotactic molecules. Gelatinase is A is MMP2 matrix metalloproteinase 1. It is constitutively expressed and activated at the surface of the cell by membrane type MMP or MMP14, and other MMPs are regulated at the transcription level. So they are controlled by signaling by the integrin, cytokine and growth factor, matrix protein binding, or tensional forces or that give mechanical stress to the tissue. Integrin MMP cooperation occurs as the MMP1 binds with alpha 2 beta 1 integrin. There is enhanced keratinocyte migration on collagen during reepithelialization. Similarly, if MT1 membrane type 1 or 2 MMP, they bind with integrin, they assist in migration, invasion, and transforming growth factor beta activation. The membrane-associated proteoglycans that are syndicane and CD44 also store and regulate the availability and activity of matrix metalloproteases. So just to summarize that in addition to the cell-to-cell adhesion and release, the matrix metalloproteinases have got other function. They may activate or inactivate the bioactive molecules such as growth factor, chemokines, growth factor binding proteins, angiogenic regulator, and matrikines that are the extracellular matrix degradation fragment with the signaling function. So their activity is basically confined to the pericellular space via substrate specificity, limited diffusion, sequestration, and there is presence of various inhibitor to these, and the key inhibitors for these MMP activity are tissue inhibitors of metalloproteinases and alpha 2 macroglobulin that is plasma proteinase inhibitor. The related proteases families that are involved in this activity are ADAMs and ADAM TS. The ADAM uh, is responsible for shedding of cell surface ectodomain, growth factor receptor, cytokines, and the ADAM-TS cleave large extracellular matrix substrates such as aggregates, von Willebrand factor. Thank you for watching this video. If you have any query, please write in the comment box.